One of the challenges in finding biological information is that very little of biological knowledge has been formalized. There are, of course, many databases out there that catalog biological data. However, none allow you to perform abstract queries. For example, you can ask for a page describing the sequence and various properties of lambda repressor, but there is no database yet that allows you to express the query, "Give me all pairs of promoter and transcription factor in which the transcription factor inhibits transcription of the promoter." The deeper we go into biochemistry, the worse this problem will be. So today, you'll need to be able to do it the old-fashioned way, by reading papers. So let me describe to you the biological literature as a roadmap. Most of the literature relevant to synthetic biology is from the biology domain. By that, I mean most of it is accessible through PubMed, and you can query titles, authors, and abstracts for phrases of interest. There is some literature outside of biology that won't be indexed in PubMed. This includes pure chemistry, physics, and related math, and non-biological engineering topics. There is a short list of commonly read general audience journals. The most prominent work is published in Nature, Science, and Cell. Nature and Science are super general in the sense that one article might be about Antarctic ice and the next about photonic bandgap materials. Cell is specific to the biodomain, but is general within that domain. PNAS is a broad audience journal like Nature and Science. The PLOS journals are specific to bio, but general within it. There are a large number of clinical biology journals that deal with topics related to human health. You will frequently encounter these journals during your literature searches since they often are talking about bacterial things and thus use bacteria-related words. However, they are rarely a source of useful data for genetic engineering. The one caveat to that statement would be around therapeutics and diagnostic applications, where these journals may provide clinical evidence relevant to the problem at hand. There are domain-specific journals where synthetic biologists publish work that isn't of sufficient general interest to be in one of the broader audience journals. Some, like JBE and NAR, have been around for many years dealing with topics related to genetic engineering. In the past few years, there has been a proliferation of new synthetic biology journals. These are the journals where work is published, but rarely are these papers what you will be going to when searching for biochemistry data. These tend to be product or tool papers, and it is very rare right now for SynBio people to publish characterization papers where they're talking about just biochemicals themselves. For subjects that are more chemical, they will often go into a biological chemistry-oriented journal. Protein engineering and tricks associated with novel assays and chemistry tricks frequently appear in these journals. When searching for papers on issues of experimental design, these could have your answer. If you're looking for data about biochemistry, molecular biology, or cellular function, these are rarely your journals. Nature Biotech, or NBT, is currently the most prestigious biotechnology journal. This class of journals deals with problem solving in the context of biological engineering. Synthetic biology often appears in these journals. Very often your project will be based on some knowledge held in one of these journals, but they are rarely the raw source of biological knowledge. Structural biology journals deal with the three-dimensional structure of biomolecules. It is also very common for these papers to contain data about enzyme kinetics and other biochemical measurements. Also, they will very often contain data about gene expression, protein mutants, and truncations. This can be an excellent source of biochemical knowledge and an even better source for structures that enable protein engineering. The raw source for most biochemical data are the biochemistry, molecular biology, and genetics journals, of which there are many. These journals have been around for a long time and support a large community of biologists. Most synthetic biology projects have some relationship to papers in these journals. This is where characterization data is most often presented. For genetic elements that deal more with cellular function than molecular function, such as promoters, transcription factors, virulence factors, and so forth, the microbiology and immunology journals are usually where your search will lead. These fields intersect heavily with the biochemistry journals since microbiology is a highly reductionist field. They try to tie observed cellular behavior down to the chemistry of what's going on in the genes that encode that chemistry. These journals are a treasure trove of biochemical and microbiological data. A variety of search tools are available which index different bags of biologically relevant documents. PubMed indexes the abstracts, titles, authors, and affiliations of pretty much any paper relevant even loosely to biology. However, not all entries in PubMed link to the actual document. When there is no link, look for the PMID number on the page. PubGet is a nice service that allows you to fetch the actual paper from its PMID. Wikipedia is an excellent source for basic information on all sorts of subjects. Most common biologically relevant chemicals, proteins, and genes have entries in Wikipedia. Google Scholar indexes a similar bag of data to PubMed, but also includes the main body of the text. Searches that you might think to do in a PubMed sometimes are better done in Scholar, but other times the larger scope leads to a lot of unrelated links. Google Patents allows you to do similar searches across the patent literature and is your best bet for accessing that space. An ordinary Google search is also effective for biologically related queries. There are many databases and ad hoc documents on the web containing useful knowledge. You just have to sift through a lot more unrelated stuff to find what you want.